Hey, it's Noah here, actually from the future doing some editing. Today's episode, actually episodes, is going to work a little bit different than the past few have. Today we're talking about biotechnology, but we got on two pretty different topics. First, we talked a lot about the tools and mechanisms and really the nitty-gritty chemistry and biology that make up past and current biotechnology. And then we moved on to more moral questions, thinking about how the future of biotechnology might impact society. We talked about things like designer babies and neurodiversity. So after listening through during my edit, I thought it more prudent to actually break this up into two different episodes, both to kind of make them more digestible and because I think they might appeal to different audiences. Someone who might not want to get into the nitty-gritty of the biotechnology might still be interested in the moral implications. So this episode you're listening to right now is the first one that we recorded and the one in which we go into the chemistry and biology of biotech. We talk about the history of biotechnology and some tools like CRISPR and directed evolution. So I'm going to let you get straight into the episode. I hope that you enjoy it.
Both you and I have classes and do research around biochemistry and genetics and cell biology, those types of things.
You're more like chemically type stuff, and I'm more
yes.
like...
So I'm a chemical engineering major.
Yeah.
So my focus is more on industrial production of chemicals, but my research is biochemistry.
My majors, I double major, are fairly specific and convoluted, and if I listed them both, if someone plugged it into Google, they could almost definitely figure out what university to go to because they are fairly specific when put together.
And I think your major has the word biology in it like 5 times.
Well, it's a double major with an emphasis, and so yeah, like 3 or 4 times there's the word biology in it. But basically, I do microbiology, and genetics, but I also focus a lot on plants, ecology, and kind of agricultural stuff like that.
Yeah, so that's where the two of us are coming from, and the way I have said it in the past, which I think is perhaps a true and a useful place to start out, this is our thesis, I guess, for the episode, is that how computers and digital technology have radically altered our world in the last 20 to 25 years, that same type of seismic shift is going to happen over the next 20 years with biotechnologies, with genetics and molecular biology and biochemistry and protein engineering. Over the next 20 or 25 years, I think there's going to be a massive change and a massive advancement in these fields that will fundamentally alter how we live our lives.
Most definitely.
How we produce everything that we consume, how we interact with medicine, how we diagnose diseases will all be radically altered by advances in biotechnologies.
Yeah, and I think something that is worth noting, but we probably won't dive into too much, is that these accelerations in biotechnology are exponentially more so because of the advancements we are seeing in other areas. Artificial intelligence, increased computing power, larger use of massive data sets and collection of data. All of that stuff, well, not within, computers and biotechnology could often seem like, oh, life and technology, they're so different, but they are deeply, deeply intertwined.
Absolutely. And the reason, I think, to understand our current tipping point in biotechnology, we have to look back at the history of biotechnology. Our listeners may not know exactly what we mean when we say biotechnology, but humans have been taking advantage of microbes to do biotechnology for a very long time. The classic thing that chemical engineers do is distillations, and historically, distillation is something you do with alcohol.
Yeah, distillation.
Yes, and brewing yeast to make alcohol is probably the oldest form of biotechnology humans have.
And it's deeply human. There's some thought that the brewing of beer and the making of bread are, in part, one of the reasons why humans came together and started the agricultural revolution. And why civilization was born was probably a whole bunch of different factors. but one of them very well could have been beer and bread, which are both based in primitive biotech.
Exactly. I mean, any fermented food, right? Kimchi, sauerkraut, yogurt, pickles, all of these things. Cheese, yeah. You make these with microbes. And it's not a very controlled form of biological production, but it's a very deep and historical thing.
All right, quick interruption. James and I, at the end of recording, realized that we talked about a lot of things without explaining them as well. So if biology isn't your wheelhouse, here is a little crash course from the both of us, just going over the basics of what we're going to talk about, just kind of as a bit of a primer. It's about 5 minutes long, so if you're not super familiar with biology, give it a listen, and I think it will help you out a lot. But if biotechnology is your thing and you don't want to listen to that, you can skip forward about 5 minutes to a time code around 11:50. Hello, it's James and Noah from the future. We're going to give you a little lesson in biology and Biochem 101. If STEM is not your wheelhouse, I'll pass it off to James.
Yeah, so we just recorded the whole episode and realized most of you probably aren't biochemistry people like we are and may not know a lot of these terms. So we want to give you a very brief crash course in what we're going to be talking about. So some of the basics. If you think about your cells as a factory, for example, proteins and enzymes are machines in the factory. And all of the nuts and bolts and pieces of metal that create that machine are amino acids. A protein is a long string of amino acids from usually between 60 and 1,000 amino acids. And any change in that string can change the machine pretty drastically, so it needs to be precise. And that is encoded for, the instructions for how to build that machine are stored in DNA. So DNA holds the information. Amino acids are the assembly pieces, all the pieces and parts that make it up. And a protein or an enzyme does all the things.
And it's important to note that we use the word protein and enzyme interchangeably. It's like a rectangle square situation. Enzymes are a type of protein. Specifically, enzymes are a type of protein that catalyze a reaction. They make it happen. So you could think an enzyme is a protein that could take two molecules and stitch them together, or two molecules and rip them apart. They're a protein that manipulate molecules.
Yes, and there are other proteins besides enzymes. Some in your body make your hair, they make your skin, they make the structure, they're in your blood. Proteins are most little pieces.
Our DNA has our information, which is then turned into proteins, and I think we can hammer it home a little bit harder than you even said. Everything about us, every phenotype, everything of how we look, even maybe how we think, is due to our environment and our genetics. And the way our genetics impact our body, our biology, is almost exclusively through proteins. And so when we talk about proteins, I think you might think of meat and beans have proteins in it, but everything, almost everything in your body is proteins, from how your brain works to how your blood works, how you see, how you feel. It's proteins. Proteins. So if we can understand proteins, how to manipulate them, how to change them, we can manipulate most body systems.
Absolutely. I mean, there are proteins in spider silk that are stronger than steel. There are proteins in jellyfish that glow in the dark. There are proteins in the human body that are excellent at tracking down and destroying bacteria and viruses. There are proteins in lobsters that can make them live forever. Proteins can do anything. And understanding the DNA that codes for the proteins and understanding the proteins themselves that do the thing are two halves of the same coin for fundamentally changing what life is and what it does.
And I think some more targeted things that we talk about, we will talk a lot about the modification of genes, genetic engineering, through a couple different mechanisms. The broadest one that life has the most experience with is evolution, or most commonly evolution by natural selection. And in a nutshell, basically evolution is you have an organism in an environment with a bunch of other organisms, and the more successful organism, the one that does better, is going to have more kids. It will mate more, it'll live longer, it's going to have more kids. And so that original organism, its genetics are going to become more common. You do this over and over and over again with new little tweaks being introduced through mutations, random changes in the genetics of an organism, and you can get big changes over time. Now, we can manipulate this by manipulating an environment to get changes that we want. This is how we turned wolves into cute dogs. We, over time, changed the environment so that it would be advantageous for wolves to be, more fluffy and cuddly. We did this to crops. We, over time, planted seeds that came from good crops, and so eventually the genes for big fruit became more common. There's also more specific genetic engineering that we'll talk about that uses a variety of molecular tools.
Chemistry.
Basically uses chemistry to change the physical molecules, and it's all about the physical molecules, but to specifically change the molecules in one organism, and that will then have an effect on how that organism behaves and interacts with the world.
Obviously, Google is going to be a better teacher than we are. It'll answer your questions more directly, because we don't know what your questions are. But we hope that this at least gives you the framework for understanding some of the rest of the conversation that we're going to have today.
Yeah. It'll at least be a good primer in that when we mention things, it won't be the first time you have heard that word since high school biology.
Yeah. On with the show.
Crops. That's another huge area where we see it, in that, people will get into this with the whole idea of, oh, I don't want to eat genetically modified organisms. Well, the sad, or not really sad in my mind, but the truth of the matter is any crop that you eat is that is through agriculture, not just something you went and grabbed out of the forest, but anything from agriculture is genetically modified through selective breeding.
Absolutely.
Corn used to be this little grass with 3 kernels on it, and now it's this freak of nature that we cultivate in mass.
Yeah. I mean, it's ironic to campaign against GMOs when you own a pit bull. That poor dog.
Yeah. Yeah.
That's a genetically modified organism. Yeah, so I think recently there's been an advancement in biotechnologies that we can artificially alter genomes, but we've been doing this for a very long time, altering the genomes of plants and animals.
The modern advancements are less about the ability to modify the genetics of a creature or of an organism. It is about being able to do it much faster, in the individual cell versus through multiple generations of selective breeding, and it's much more targeted and specific. In the past, we could, over many generations, take a type of fruit and only plant the seeds that were from successful large fruits, and very slowly and gradually get larger and larger fruits. And now we might be able to achieve the same result of taking a small fruit and making it bigger, but instead of doing that over large periods of time through selective breeding, we do it in one generation through targeted genes. So the results could be very, very similar. It's just a faster and more specific process.
Absolutely.
So in some ways, the genetic engineering that we're doing now is more controlled and more understood. So if people want to be afraid of something, things can always go wrong, but what we're doing now is targeted and much more understood than just throwing it to evolution.
Yes, absolutely. Thinking about our current tipping point in biotechnology, however, I think the change from doing, large-scale selective breeding to specific genome editing, such as making salmon that have tomato plant genes, for example.
Or rats that glow in the dark.
The types of advancements that have allowed us to do that shift have happened in the last 100 years. For example, insulin is a protein in mammals that has been hugely important in medicine for treating diabetes. Isolation of insulin has been available for less than 100 years. It was 1922, I believe, I looked it up this morning, that insulin was first isolated and administered to patients. That's a fundamental change in biotechnology, where we go from working with organisms as a whole and trying to push for bigger cows to being able to take a cow pancreas and remove very specifically the protein that we need to treat diabetes.
Yeah.
In the 60s, Watson, Crick, and Franklin, three scientists, discovered the structure of DNA, and we were able to more specifically manipulate that with new laboratory methods. In 1983, Kary Mullis developed polymerase chain reactions for DNA.
PCR.
Which is how COVID testing is done. Yes, PCR.
The effective COVID tests, the vast majority of them are through PCR. And PCR is basically just a way in the laboratory to amplify genetic information, DNA or RNA. Basically, you put in a piece of DNA, and polymerase is an enzyme that duplicates it. You put in a bunch of nucleotides, the building blocks of DNA, and run it through a couple different heating and cooling cycles and just let it go, and you will magnify the amount of DNA that you have.
Exponentially,
Yeah,
unbelievable.
because every
amounts.
cycle you do it, you'll basically heat it and cool it, and every time you heat it, it doubles.
Yes, and it's a very precise system as well, highly precision, high precision. In about the turn of the 21st century, around the year 2000, the Human Genome Project was an open source project that sequenced the whole human genome for the first time. And so these types of discoveries, understanding how to isolate proteins, understanding the structure of DNA and being able to manipulate it with PCR and other tools, having a mapped out human genome, all of these are foundational information in the case of the Human Genome Project, and foundational lab mechanics such as isolation of proteins and PCR. These very foundational lab mechanics that have been understood and improved over the years to be a good foundation for what the modern shift in biotechnologies are, which I think is information-based, which you alluded to earlier.
Yes. An analogy that I have heard for many types of technology, but specifically for biotech, is that think of an iPhone, right, or any sort of smartphone. A society being able to build one iPhone, neat. You can build a spaceship, but you can build one, and it's a cool science experiment, but it's not as groundbreaking. But when most people can own a smartphone, or when you can send a spaceship into orbit every month, now that is when something becomes revolutionary. Because one iPhone is a neat science fair project, but a whole country with a smartphone in their pocket, that changes everything.
It's a fundamentally different society.
Yeah, and so with biotechnology, being able to edit the genome or the genetics of an organism, or being able to duplicate DNA, it's neat on its own, but when you have processes like PCR, and especially advancements in like PCR machines, duplicating DNA, easy. We did PCR in high school, whereas, 20 years ago, 30 years ago, not a high school science experiment.
Absolutely.
Or with CRISPR and other modern genetic engineering techniques, you can accomplish things that 20, 30 years ago would have taken thousands of dollars in a state-of-the-art lab with $300, a little bit of know-how, and a garage.
Yeah. I think the cost especially is a big thing to understand. With this, biotechnology is gradually growing in scale and in size, and that effect on cost is immeasurable. I'm doing research this summer, and I needed to order several thousand base pairs of custom DNA. Which
is intense. Like, I think for
yes.
as STEM majors and people in the field will kind of get that. But for anyone who's not familiar with this type of stuff, thinking about, you need this molecule. You're basically having a company make a molecule for you that you need every little bit of it to be super specific, and it is crazy the detail you need.
Yes. I mean, it's like writing a short novel and you cannot have a single typo. And you're not writing it on a page, you're assembling it out of molecules. And to get more than 2,000 base pairs of custom DNA cost my lab $350, which is an insanely low price for something that precision.
Yeah.
And so this is the kind of scale that is just starting to be possible, that is going to fundamentally change how we interact with biotechnologies.
Do you know how companies do that? A bit of a tangent, because I don't.
I believe it was an optional lecture in my biochemistry class this semester, actually, I believe, covered it. Basically, you anchor one end of the DNA chain to something solid, and there's a cycle of 4 or 5 reagents that you immerse this thing in. So it's in a tank on a structure, and you first fill it up with something that will bind your first base to the structure, and then you keep a protecting group on the other end of the base, so you don't get 2 of them sticking together.
Yeah.
And then
And then, something along the lines of and then maybe you remove the protecting group or whatever, and you dunk it in the next one that you need. So it's like making a candle or a gobstopper or something, where you just dip it in every time you need a new base. It's like making a dip candle. That's intense, because we're talking thousands of dips.
Yes. I believe that method is only good for up to about 200 base pairs, and after that you have to stitch together some smaller ones. Perhaps what is more a better way to think about it is it's like putting layers of frosting on a cake. Another good analogy. And you have to switch out between all the different types of frosting and clean out the tip of the bag and that sort of thing.
And while I don't obviously I just heard from James about how this process works, but from my understanding of kind of, biotech and this type of stuff, I wouldn't be surprised if this type of procedure, say, give it another 20 years, and that is a long time in this industry at this point. 20 years, long time. Maybe even 5 years or 10 years. I wouldn't be surprised if you could end up with a DNA printer of sorts. A machine that would just, you type in the base pairs that you need, and robotically just does it for you. You leave it running in the lab overnight, you come back in the morning, boom, there's your custom DNA. And the first one of these things, would probably be very expensive, but you give it 20 years or something, I don't know this process, I don't know how feasible it actually is, but I would be shocked if that was impossible, or if that was even, prohibitively challenging.
Right. And so I think one fundamental change that's happened is this change in scale, right? Now, biotechnology is cheap because a lot of people have started doing it. And getting custom DNA is not expensive or difficult. We went online and ordered it like it was Amazon, right? I think the other fundamental change, which you alluded to earlier, is bioinformatics. This goes back to the Human Genome Project. For a very long time, we were wandering around in the dark in biotechnology. There is so much, the information that's out there is astronomical. Unbelievable amounts of information that a human could never hope to parse through on their own.
No. And that is where enhanced computing, I hesitate to use the word AI because that's not well defined, but computer systems that can understand large data sets and interpret them in ways that are not directly hard-coded into them are becoming, think of IBM systems, or even the Google search engine, or the YouTube suggestion algorithm. We interface with these type of softwares. It's hard to programs, bot networks. It's hard to pin down a good definition, especially from someone who does not come from a computer background, which I do not. But it's becoming more and more available that you can take a huge data set, chuck it into a system that you don't even really understand yourself, and it just spits you results back. And that's becoming more and more common.
Yes. So my lab is about protein engineering proteins. And we read a paper this past spring about machine learning for protein engineering.
Machine learning, that's what I was looking for.
Yes. And traditional protein engineering requires a lot of requires a lot of expertise on the behalf of the protein designer, and it takes a long time to test all of your results. And this one paper said, we made a machine learning algorithm that if you just tell it a little bit of information about your protein, it can do the intermediate half a dozen steps for you and get you pretty close to your result, 80% of the way there.
Wow.
And it saves you, 6 months of work, and it produces results that are more or less comparable.
And give that a few years, and those type of systems won't be more or less comparable. They will be exponentially better than humans could ever aspire to be.
Absolutely.
Because, computers can think a zillion times faster than we can. And they don't get tired. And they don't slack off. And they don't fudge the numbers.
And they don't make mistakes, Machine learning models sometimes make mistakes. But
those are,
that's
those are the mistakes of their engineers.
Yes. That's one form of bioinformatics, is understanding how proteins work in kind of a forward-looking sense. What is really interesting is my research does bioinformatics looking backwards.
Yeah.
So some of the work I do is called Ancestral sequence reconstruction, which is really cool. You can take a lot of species or proteins from species that currently exist, modern proteins, and we have a decent understanding of how proteins evolve over time, at least in a short time span. And from there, you can feed existing proteins into a computer algorithm that will cluster the most similar ones and find what is the most likely common ancestor that mutated into these modern similar proteins. So if you have modern proteins that differ by a few amino acids, you can back calculate what their common ancestor is, and you can join that common ancestor to its next door common ancestor. And before you know it, you have a whole family tree for all the proteins, not just that exist today, but have ever existed. And obviously there's some uncertainty. We're guessing at random events.
They're theoretical proteins in a certain sense.
Yes.
They are proteins that could have, if they existed, would make the family tree of proteins, as you put it. It would make sense for that. In reality, maybe historically, it looked slightly different, but for your research and purposes, it's semantics at that point.
Well, yes. So for my research purposes, it doesn't matter, because we want these ancestral proteins because they're useful, not because they're accurate to the true ancestors. Some other scientists are actually interested in what the true ancestor proteins are, and although I am by no means an expert in it, their work indicates that these programs are pretty accurate. They have observed, for example, thermostability in proteins that models the heating and cooling of Earth over time that we know from geology.
Unpack that for someone who's not a STEM major.
Yes, so through geology and other sciences, we know more or less how hot or cold the Earth was over the last 3 billion years. And proteins, as a chemical, have a certain temperature range that they can operate in, and for humans, proteins like to operate at 98.6 because that's what our body's at.
No.
But bacteria often just float around in the ocean. They don't get to keep a body temperature. And so the temperature range that a bacterial protein will operate at is the temperature range of the ocean. So because we know how hot the ocean was 30 million years ago from geology, we can correlate that with how thermostable are the ancestral proteins that we get from ASR. Like I said, I'm no expert in it, but people who are experts in it indicate that many ancestral sequence reconstruction efforts make sense for the actual ancestral conditions of the proteins, that it's not just a guess, but it's pretty accurate.
That's so clever.
Yeah. Science is cool, man.
I love it when scientific disciplines that seem so far apart come together to do something. I love imagining you've got this, PhD protein engineer, software engineer guy who's like, oh, okay, so I need a guy who's like a geologist. I need a geologist. I need to consult with a geologist for my protein engineering.
Yeah.
So I think something that gets a lot of buzz nowadays is CRISPR. And for me, it's something that I've been paying attention to for a little while because I want to be a geneticist. But I think it gets thrown around a lot, and while it is revolutionary and groundbreaking, there is a certain level of understanding that needs to be had about it. It is not a magic tool or some silver bullet where you take an organism and you just CRISPR it and turn it into whatever you want. But it is something new and something different and special. So CRISPR is an initialism, or I believe it's technically an acronym because you say it.
Yes.
For clustered regularly interspaced palindromic repeats. Now, that is not a single object. It's more of a system, and it's not a system that we humans came up with. It's a system inside of bacteria. We didn't make CRISPR. We discovered it and then have hijacked it for our own purposes. Basically, bacteria have, since bacteria and viruses have both existed, viruses infect bacteria. And bacteria don't want to be infected by viruses because that's bad for them.
And they don't have an immune system to fix the viruses.
No, because they're one cell and they're much simpler than we are. So at some point, they evolved a way to develop some sort of immunity or resistance to viruses that they or their ancestors had already encountered. And basically, to simplify it a decent bit, sometimes when bacteria that have this kind of system in them, if they get infected with a virus and then survive it, sometimes, if they've got the proper things in the right place and the conditions are right, a portion of that virus's DNA or RNA, but will be their DNA into the chromosome of the genetic information of the bacteria will be incorporated and stored there in a way that's not going to hurt the bacteria. And then the bacteria has a special protein called Cas9. So when you're talking about CRISPR in terms of its use for genetic engineering, it would probably be more correct to say CRISPR Cas9.
As a system, the pair of them. Yeah.
The system. CRISPR Cas9 is the system because Cas9 is the enzyme. Basically, they have this enzyme which can read or kind of recognize and take the piece of virus that they have stored within them and go around the cell and look for that same piece. And if it finds that same piece, it means it's found virus DNA and it will cut it. That's good for the bacteria because basically they've got this catalog within them of, hey, if you see this DNA, get rid of it because it's going to mess you up. But we can take advantage of it by basically taking the Cas9 protein, and instead of giving it just, bacteriophage DNA, we give it whatever DNA that we want to cut, and it will find that specific piece of DNA and cut at that specific point. The reason that's so revolutionary is because of its specificity. We can give this protein a very specific sequence of DNA, and it will cut at that specific point, and we will then know that there's a cut there, and we could basically give it a gene of some organism, set it loose, it goes snip, 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 cuts that out, and then we take a different version of that gene, put it in there with some other enzymes that repair the DNA, and now that organism has the new copy that we want of that gene. And that's a drastic oversimplification, but that's the important parts of it. The important part to understand is that CRISPR is in fact revolutionary, but it is not a magic bullet. It is still complicated and takes work. It's not something we created. We discovered it in bacteria and then use it for our own purposes. And it's important because it's specificity.
Yes. So in the past, we have been able to insert DNA into cells sometimes, but it's usually not where we want it to be. CRISPR Cas9 gives us a pair of very precise scissors so that we can do the cut half of a cut and paste. And I think, again, the precision is the key, right?
Yeah.
That these are things we've known how to do for a long time, but now we can do them exactly how we want to do them.
Also, CRISPR, it's complicated science, but compared to some of the past methods, like there were methods before CRISPR to do this type of procedure, but those were expensive, not as good, much more challenging. But CRISPR, it's still relatively new and people are improving on it, but you can buy online as just a normal person a CRISPR kit where you can do, at-home sciencey experiments for a couple $100 as a normal person.
Yeah.
And labs, universities, you can just use CRISPR. You can teach a student to use it. It's not exceedingly challenging to learn how to do. It's not exceedingly expensive, and you can get pretty good results with adequate effort.
Yeah. And in fact, that was such a successful discovery, such a good discovery. It won the 2020 Nobel Prize for Chemistry just this past week. Interestingly, in my field of protein engineering is a woman named Frances Arnold, who is a chemical engineer by training, but does biochemistry, like me. And before this announcement of the CRISPR Nobel Prize, Frances Arnold was the only American woman to win the Nobel Prize in Chemistry. She won in 2018 for her work in directed evolution. And what Frances Arnold has done is really incredible. Her research goal, her big picture research goal is that we can solve a lot of problems with proteins and enzymes that we could never do with kind of standard traditional chemistry. So when it comes to trying to make chemicals or do chemistry, nature has 3 billion years head start on us. There's no way that our lab mechanisms are ever going to catch up. And so using natural enzymes is such a better way to do it. The problem is natural enzymes don't always do what we want them to do. So Frances Arnold, in her 40 years of research, has developed a number of methods for highly directed evolution. For example, there are no enzymes in nature that bonded carbon to silicon as an enzyme, enzymatically binding carbon to silicon. No enzymes did that in nature until 2004, when Frances Arnold's lab engineered a brand new enzyme that could bind carbon to silicone in a highly specific and useful way. Carbon-silicone bonds are really useful in a number of products like plastics and hair gels and soaps and you name it. I mean, if it's some sort of paint or plastic, it probably has carbon-silicone bonds.
Yeah, well, and also, I don't know really anything about her work, right? But just thinking of the broader thought of biotechnology, if you want to start talking about body-safe implants, silicone is used in all sorts of electronics, and carbon is used in all sorts of organic molecules. So even towing the line between biology and technology, carbon and silicon would probably have to be intertwined for that.
Absolutely. Another thing that Frances Arnold's lab did is they made a bacteria that produces jet fuel.
What?
So
What? That's incredible.
Yes. One of the biggest sources of carbon emissions from petrochemicals is jet fuel, because it takes so much energy to get an airplane off the ground, and we have to pump that energy out of oil. Previously, there were no organisms that made
jet fuel. That's not a shocker.
Compounds for jet fuel. Yeah. Why would a bacteria need to make airplane fuel?
Yeah, I mean,
right?
just go down to the local pond and find yourself some jet fuelless spirulina.
No, it's not a thing that existed in nature. But Frances Arnold's lab used directed evolution to very rapidly and drastically change the genome of some small bacteria, and they now produce jet fuel.
Do you know more about their methodology in terms of what you mean by directed evolution? Is that, not the manipulation of the, not going in on the molecular level? Is this just changing the environment in such a way that it would push it in that direction?
Yeah, so let me talk about a good example here. What they did was they did evolution in a controlled sense. Her seminal work in 1993, as an example, that really won her most of her notoriety, was a literal standard lab bacteria. It wasn't anything special, with a very specific enzyme that is used to cut up things outside of the bacteria. So the bacteria excretes this protein, this enzyme, and it cuts up things outside the bacteria.
For like digestion, I would imagine.
Yeah. And obviously bacteria are usually floating in water
Yeah.
of some kind, and so this enzyme works in water.
Yeah, it's like if you were to, vomit on your food to like digest it a little bit and then lick it back up.
Yes, that's what bacteria do. However, a lot of industrial reactions don't take place in water. And this enzyme that is good at breaking things down would be useful for industrial reactions. So she took a bunch of these bacteria and dumped them into industrial solvent. And then made the environment such that they could only eat if they produced an enzyme that worked in industrial solvent.
Fun.
And the vast majority of those bacteria died because they can't live in organic solvents.
But as long as some live.
But they found a few colonies that lived just a little bit, that didn't die quite as much, and they took those colonies and they allowed them more time to mutate, and they threw them back in organic solvent. And this time they got a bunch more that worked a lot better. And over time, they were able to really heavy-handedly force evolution on this poor little bacteria to make a version of the enzyme that functions in nasty industrial solvents instead of in water.
So it's like, this reminds me of antibiotic resistance. So it's basically taking this process of dousing a bacteria in a compound extremely harmful to it, and if only a few survive, well, then they become the next generation. That is exactly how antibiotic resistance occurs. And so it's basically taking the process that forms antibiotic resistance and using it for something good and useful, which is fascinating, and I had never thought of, but that makes so much sense.
Absolutely. From this method, from the very first enzyme that showed the tiniest little hint of being able to do this through natural variation, she increased its functionality 256 times over.
Dang.
Which is a lot. Other things that she's done that are interesting is enzymes that make cyclopropane. So a propane is a 3-membered carbon, and it is a really nasty chemical, really difficult chemical, when you bend it back on itself and make a triangle out of the carbons. It's not useful for a biochemistry perspective. There's no reason to have cyclopropane in nature. But she engineered bacteria that could make cyclopropane, which is useful for a variety of industrial reactions, including pharmaceuticals. And she's done this with a bunch of different things. It's really cool work, this directed evolution.
That is so cool. That's fascinating.
Yeah. I mean, it won her a Nobel Prize. It's great work.
Yeah, that's incredible.
Her end goal, like the jet fuel bacteria, is that nature has incredible tools for doing chemistry. And as a society, we don't need to be dependent on natural gas and coal and oil and tar if we can use bacteria to do the chemical reactions for us, to build it up from small things instead of breaking it down from big, long oil chains.
Like doing metabolism backwards, basically.
Yes. Imagine if you could grow plastics using a giant pond full of bacteria instead of needing to pump oil out of the ground. That would be a great way to do carbon fixation if you can fix carbon into plastic from the atmosphere. That could just solve global warming.
Holy crap! Yeah, I mean, if you set up an environment where you pump the CO2 into a liquid, bubble it through the liquid, just bubble. If you could just set up basically think of a water treatment plant where you just bubble the atmosphere through a vat of water, and the bacteria in it selectively take the CO2 and other greenhouse compounds out and turn them into something useful. Yeah, that'd be pretty damn nifty.
Yeah, it'd be incredible. And so this won Frances Arnold the 2018 Nobel Prize for Chemistry. And it's some biotechnology that I think is going to be revolutionary. This is the kind of work that's going to change our lives over the next 20 years.
Well, especially when we combine everything we've been talking about. When you combine further understanding of evolution, and then maybe you hit a little bit of roadblock, well, you hit a little roadblock, and we've got these targeted tools where especially when you say, oh, that sounds really complicated and hard to understand, well, we don't have to understand it all. Our computers have to be the ones who understand it. So you could end up in a system where you have an end goal and you feed into a computer, you give it a starting bacteria, but it doesn't just have that starting bacteria. It has a database of all genes we know, and you could just say, hey, give us a protocol where either through evolution or through inserting genes, we get the results we want, we get the proteins we want, and it's not infeasible to say that you could have an artificial intelligence in 10 years and not like some Skynet, takeover, not some I just mean like a really good computer program that can suggest pathways to manipulate organisms into producing proteins that are novel or that we want.
Absolutely. And so the first step in the process of doing really cool biochemistry work of, the information, right? I have an end goal in mind. What does that look like as a biomolecule? That type of informational work can now be solved through bioinformatics and high-powered computing.
Yeah.
And the lab tools for making this information we have about what we want to make, the lab tools for making that information into a physical reality, like CRISPR, like directed evolution, have seen massive improvements over the last 20 years. And I think a combination of massive improvements over the last 20 years and improvements in terms of scale and cost have really put us at a tipping point currently towards good biotechnologies.
Most definitely. It's an exciting time to be in genetics / biochemistry.
Absolutely.
It's one of the reasons why I'm going into the field, because I think we are at a point where
Me too.
I think we both are at a point where the field is exciting, and we know that if we are working in the field, we will be at hopefully the golden age of genetics and biochemistry, where we have the tools at our disposal to make revolutionary change, but we are at the point where we are the ones doing that revolutionary change.
And this type of work, it's almost limitless. It feels almost limitless in what we could do. Imagine if there are bacteria that just grow anti-cancer drugs in a vat, and it costs like 25 cents because you just throw some sugar and water and bacteria together and they pump out chemotherapy.
Or imagine if instead of landfills, we just had big vats full of a whole cocktail of, we basically engineered an ecosystem that was just really good at breaking down our waste. If instead of landfills, we just had pits of yeast that eat plastic and bacteria that break down whatever other shit we throw in there. And in the end, we have a biologically inert compound that is not a hazard.
Or they don't even need to break it down. We could feed them our trash and they could turn it into cancer drugs or jet fuel or we could plastic.
Or, we just feed them our trash and maybe they synthesize it into, maybe some like recycling efforts. Oh yeah, throw your food scraps in this bin. They'll bring it to the microbe dump, and then they'll spit out synthesized meat and we'll eat it tomorrow.
Exactly. I mean, these are the types of crazy things that could be possible with our biotechnologies. I think we've mostly talked about industrial chemistry because I'm a chemical engineer and that's what I'm interested in. But the medical applications here are incredible as well.
Yeah. Well, and the thing is, I think part of the reason why you end up, when you start talking about medical things, and I think we will get into this soon, you start having different conversations because you talk a lot more about like, everyone's like, break down our garbage with yeast, awesome. But you get into a lot more moral gray areas, and you end up talking a lot more about systems and political powers and economics when you start talking about human lives on the line.
That's true.
At least in a direct way. Climate change, human lives are on the line, but not in a direct way. But you want to start talking about, well, hey, having access to medicine that could not just treat diseases, but eliminate them before they happen. That's some heavy shit.
Well, I think more of those ethical concerns come from the genetics standpoint.
Oh,
which
most
is where
definitely.
You're coming from. From my perspective,
people love treatments.
Yes. I mean, proteins are all about treating things that already are problems. Some of the research that my professor in my lab did to get his PhD involved super high precision cancer screening for aggressive cancers that are hard to detect. And by using some different proteins and using some novel methods, he got very high precision results to find, tumors that are usually difficult to detect through protein engineering by designing a protein that sticks to cancer. And there's no reason to stop there. We could design proteins that just lyse cancer cells that pop them open.
See, and then you have the futurist geneticist. I'm not saying that's me, but
Yes.
A potential answer to that could be, why design proteins to fix problems when you can just design people to not have them? Hey, it's Noah again. Thank you for listening to this episode of In My Experience. If you enjoyed it, do remember that we actually have 2 episodes out on this topic. The next one is about the ethical and moral implications of biotech, which I think we kind of hinted at the end of this episode. It's actually just a continuation of the same conversation we were having, and I'll probably even include a little bit of overlap at the start. So if you are enjoying the conversation you just listened to, definitely go check out the next episode where we're going to talk about the moral implications of the technology that we just discussed.